Welcome back, everyone. In this lecture, we will discuss cellular respiration. This chapter will be applying the concepts we've learned in chapter 1 through 6 and is going to dive much deeper into the inner workings of the cell. If you feel uncomfortable with the material up to this point, it would be beneficial to you to review those concepts before tackling this chapter. A good place to begin in this lecture is breakfast. The examples here, I'll provide you with some of the nutritional macromolecules of carbohydrates, lipids or fats, and proteins. The fourth macromolecule, nucleic acids, are not of nutritional importance and something our cells synthesize all on their own. However, carbohydrates, lipids, and proteins can all be used to provide energy and building blocks for our bodies. This process of converting the bond energy of biomolecules into energy that is usable is a major component of metabolism. The second would be to use this energy to do work for the cell. So, in essence, we are converting potential energy into kinetic energy. In the prior lecture, we discussed how organisms harvest chemical energy. This is namely done by breaking the bonds between atoms. Keep in mind that not all chemical bonds have the same amount of energy as others. For example, carbon-hydrogen bonds release more energy when broken than their carbon-carbon or carbon-oxygen counterparts. Therefore, the rearrangement of bonds between different atoms can release energy that can be then harvested by the cell. You'll come to see how a lot of cellular respiration is about converting the chemical bonds carbon has in a molecule, such as the sugar, into CO2 bonds, and in the process, gathering energy from sugar to be put to work later. While energy stored in sugar is fine, it takes specific enzymes and complex processes all by itself to release the energy. This simply won't suit the wide array of activities in the cell that require energy. Instead, the cell has a chemical called ATP that can more easily release its energy around the cell. Therefore, the cell has devised an elaborate process to convert as much of the chemical bond energy in sugar to ATP as it can. And we'll discuss how the cell does this through a process called cellular respiration. Cellular respiration can be broken down into four main steps. These steps occur in either the cytoplasm or the mitochondria. The first step, glycolysis, occurs in the greater cytoplasm of the cell. The other three steps, pyruvate oxidation, citric acid cycle, and oxidative phosphorylation, all occur in the mitochondria. Cellular respiration is really the chief function of the mitochondria, and therefore its anatomy is set up in such a way to try to maximize this ability. We've shown a few microscopy images of mitochondria, but this figure is a better representation for its structural features. The mitochondria has two lipid membranes. Starting from the inside out, we have the mitochondrial matrix, which is to the mitochondria what the cytoplasm is to the cell. Next is the inner membrane, and then the outer membrane. Between the two is called the intermembrane space. The inner membrane has a very unique membrane structure with numerous folds called cristae, which can be seen in the microscopic image dyed pink in color. We are going to be covering the steps of cellular respiration, and you will notice a theme throughout, and that is oxidation of organic compounds in order to extract the energy from their chemical bonds. Recall that atoms or molecules that are oxidized lose electrons, and those that are reduced gain electrons. In this chapter, we'll focus on how sugar is oxidized to release the energy stored in their chemical bonds. Here's an example of sodium, abbreviated as Na, being oxidized by iron, abbreviated as F, and iron reduced by sodium, is shown here. Now, this is a rather simple example, and the process of cellular glycolysis deals with organic biomolecules and is a much more complicated process. Before we get into it, let's just add a note to keep in mind that will better help you follow the steps. When we see a loss of an electron from a molecule, it will come along with a loss of a proton. As a reminder, a proton and electron together makes up a hydrogen atom. This is where it gets a bit confusing because we have learned about dehydration reactions, which are chemical reactions where a water is lost. However, these are called dehydrogenation reactions, where a hydrogen atom is lost, not to be confused with dehydration, like when a water is lost. As an example, here is a fatty acid. Within the fatty acid, there are these carbon-hydrogen bonds where carbon shares one of its electrons with a hydrogen, and a hydrogen shares its electron with a carbon. Now, a molecule called FAD interacts with the fatty acid, dehydrogenating two hydrogens. Here we see the FAD picks up two hydrogens and the two electrons. Now, what does carbon do with its lone electron? Well, after taking their electrons, we see here that it's leaving only the carbon's electrons behind. And what the carbons do is they share their lone electrons together. So now we've restored the total valence electrons for the carbon to 8. So each one of those carbons still has 8 electrons, counting the electrons it's sharing with its neighboring atoms. As a reminder from the biomolecules chapter, here we converted this fatty acid from a saturated fatty acid in which it has all of its carbon bonds are saturated with hydrogens to an unsaturated fatty acid with the carbon double bonded to another carbon. Checkpoint 1. We will focus on the metabolic breakdown of sugar molecules. Sugars are the monomers of what biological macromolecule? A. Lipids, B. Carbohydrates, C. Nucleic acids, or D. Fats. Okay, let's take a step back and look at the overall picture. Here's everything condensed into a single chemical reaction. Take a moment to reflect on what glucose is. It's a carbohydrate. A carbohydrate is a carbon that has been hydrated. In a carbohydrate, for every one carbon, you have one water molecule, or you have H2O, two hydrogens and one oxygen. So here we have, we've mostly been showing this as a ring image, as in the previous checkpoint here. But what we have here is if you take all the carbons, hydrogens, and oxygens, you get 6 carbons, 12 hydrogens, and 6 oxygens in a glucose molecule. If you reduce those down, divide by 6, you get 1 carbon, 2 hydrogens, and 1 oxygen. Again, a carbohydrate. Basically, the cell consumes glucose or carbohydrates with the use of 6 oxygen molecules, and oxygen occurs here on Earth as O2. So there's actually 12 oxygens altogether, 6 oxygen molecules. And the cell oxidizes the sugar with those oxygen molecules to make 6 carbon dioxide molecules, 6 water molecules, and stores the energy released from the sugar bonds as ATP for later use. You eat glucose, you inhale oxygen, you breathe out the reduced or the oxygenated glucose as carbon dioxide gas, and you produce water, and you're mostly water. And you need ATP, you need energy, so you create ATP. Here's ATP. Its full name is adenosine triphosphate. Why? It's made up of an adenine nucleic acid, a ribose sugar, and three phosphates all attached in a row to the ribose sugar, called a triphosphate group. These phosphates store a lot of energy in an easily accessible manner. So we have nucleic acid attached to ribose that is attached to phosphate. And this should ring a bell about another biomolecule we have previously covered. And so, checkpoint two, what does the structure of ATP closely resemble? Sugars, carbohydrates, and fats store a large amount of chemical potential energy, but that energy is not easily accessible to the cell. However, ATP can be easily liquidated into energy, and that's what makes it so useful for cellular activities. The equivalency here would be storing, assets such as commodities of gold, diamonds, maybe cryptocurrencies, whereas having cash can be something that you can easily use as a monetary system. The oxygens shown along the bottom of each phosphate in the triphosphate group have an extra electron, which is why we are denoting those oxygens with a minus sign to denote that extra electron and to show that they are electrons and to show that they are negatively charged. These oxygens cause what is called electrostatic repulsion between the phosphates because of these charges, and therefore make it easy to break an electron bond and release the energy of the bonds between phosphates. So highlighted in red, the bond between uh, one phosphate group to the next, that bond can be broken at the end quite easily to release the energy. Fully charged version of this molecule has all three phosphate groups. When the cell wants to use an ATP molecule, it will break off one of the phosphates, releasing the energy. As a result, the adenosine triphosphate becomes adenosine diphosphate, or ADP. So watch out for that. Another way we can think of this as energy flowing from Another way we can think of this is as energy flowing from long-term potential energy in carbohydrate, lipid, and protein chemical bonds to short-term potential energy in ATP. And then finally being turned into kinetic energy to do work. Here's what is called a metabolic map of a cell. Now, this is not an all-encompassing metabolic map. There are a variety of reactions that molecules can take to either be broken down for energy or built into other molecules through a chemical reaction. And many of these pathways shown here, they're made up of many steps along the way, as we'll soon find out. And down the middle here, this backbone here is what we're going to be focusing on. So this highlighted region here is the carved out region of cellular respiration, where the cell takes up sugar and goes through glycolysis, pyruvate dehydrogenation, the citric acid cycle, and finally oxidative phosphorylation. And this here is an overview of where each step of the process takes place. The image represents a broad overview of aerobic cellular respiration. At the top of the image, we can see a cyclic ring of glucose entering the system. As we follow the pathway downward, the image shows the location of the four steps within the cell when hydrogens are being harvested and the production of ATP. We'll start at the top with glycolysis. Looking at the image, we can see it occurs in the cytoplasm, produces ATP, and siphons off protons and hydrogens to make a molecule of NADH. So as we go through each one of these steps, there's going to be it's going to appear like there's a lot to keep track of. But what I really want you to focus on are these 4 items. Every step you need to understand where the step in the process takes place in the cell, what is the starting material, and what are the ending materials? And additionally to this is CO2 being generated? And if so, how much? The third thing to keep track of is how much ATP is made in this step. And lastly, number 4, how much NADH or NADH-like compounds are made. We'll get into that a little bit more detail when it comes up. So we have our list. Now let's apply it here. Glycolysis is the metabolic pathway where glucose is taken and broken down into 2 pyruvic acid molecules, sometimes referred to as pyruvate. Okay? Remember that glyco means sweet and lysis means to break apart. So our starting material is glucose and it's going to be broken apart, and the ending material is going to be 2 pyruvates. There are actually many substeps in glycolysis. This is just the overall reaction, start to finish. But this slide shows the overall pictures, and our glucose molecule has 6 carbons. They're not all drawn out here in these rings, so don't get too confused. In the ring, each point in that cyclic ring where there's not an oxygen listed, that's going to represent a carbon that goes there. And there's actually hydrogens there as well that aren't shown, but we really want to focus on the carbons here. Like I said earlier, we're counting carbons. So um, in that ring, there's 6 corners. One of them is designated as an oxygen. So there's actually 5 carbons in that ring. And then off the top there on that flag, we have another carbon. So there's 6 carbons there. Okay. And then the end product is 2 pyruvates or 2 pyruvic acids. And those are 3 carbon molecules. See, there's 2 points there. And at the very tippity tip on the left-hand side of those, the way those molecules are drawn, there's another carbon there. Okay, so there's 3 carbons in pyruvic acid, and there's 2 of them, and there's 6 carbons in glucose. So as we can see, we didn't create or destroy any carbons. It all adds up. All the math adds up in the end. From this step, let's get back to the main big picture here. From this step, the cell has gained 2 ATP, and 2 oxidations have occurred because 2 NAD pluses took a proton and electrons to form NADH. If you look in the top right, it summarizes our scoreboard here. So far, we have 2 ATP and 2 NADH. Okay. Let's pause to see what role NAD plus and NADH serves in all of this. This abbreviation stands for nicotinamide adenine diphosphate. This molecule is an electron carrier. It can be reduced by oxidizing another molecule, or it can be oxidized by reducing another molecule. And that might sound a little confusing, but remember from last chapter, anytime a reduction occurs, there's a partner in that, which becomes oxidized and vice versa. In order to give an electron, someone has to receive an electron and vice versa. So ATP's job is to store energy released from bonds, and NAD plus's job is to facilitate this process by oxidizing compounds along the way. And it'll have a very special purpose in the end of aerobic cellular respiration, which we'll see later. Like ATP, NAD plus is also an energy storage molecule. ATP is the charged version of the molecule, and ADP is the uncharged version. Remember, that ATP has energy and the ADP does not. NADH is, you could think of it as the charged version of the molecule. It has the extra electrons, right? And NAD is the uncharged version. It's lacking the electrons. The roles are very different though, okay? We want you to keep in mind ATP is for energy purposes. Nicotinamide adenine diphosphate is for moving electrons around. Okay, checkpoint 3. How many pyruvate, ADP, and NADH are produced from the glycolysis of one glucose molecule? 1 pyruvate, 1 ATP, and 1 NADH, or B, 2 pyruvate, 1 ATP, and 1 NADH, or C, 2 pyruvate, 2 ATP, 2 NADH, or D, 1 pyruvate, 2 ATP, and 2 NADH. So that covers glycolysis. Now, if we follow pyruvate down the metabolic pathway here in our little image, we see pyruvate oxidation is next. And this occurs in the intermembrane space of mitochondria. In pyruvate oxidation, you guessed it, pyruvic acid is oxidized and it becomes acetyl-CoA in the process. Its oxidation gives off a CO2 for each pyruvate. Remember, there's 2. And therefore, this 3-carbon molecule becomes a 2-carbon molecule as acetyl-CoA. Once again, another NADH is produced per pyruvic acid. Reviewing our scoreboard now, we have 2 ATP, that remained unchanged, and 4 NADH. We gained 2 NADHs because we sent 2 pyruvic acids through pyruvate oxidation. Again, moving down the metabolic pathway, we get to the citric acid cycle, or as it is sometimes referred to, the Krebs cycle. This metabolic pathway occurs in the mitochondrial matrix. It further oxidizes our acetyl-CoA, and in the process gives off ATP, NADH, and FADH2. And here's a closer view of what this cycle looks like. This one will show a little bit more detail, but remember, we're going to be interested in the starting material and the end materials. Okay, that's the important thing to pay attention to here. Okay, so acetyl-CoA reacts with a molecule called oxaloacetic acid, seen as it with that arrow that merges together. And it synthesizes a chemical called citric acid. It is treated through a series of enzymatic steps until the final product is back to where we started again, is oxaloacetic acid. This is why it is a cycle. As previously discussed, we're not interested in knowing everything here, but only those items on our list to keep track of. So our starting material is acetyl-CoA, a 2-carbon molecule. We have 2 of them. And we generate CO2 per carbon per acetyl-CoA. So that's the end of our oxidation steps for our original glucose source. We've turned all of our 6 carbons into 6 CO2s now. Okay. Recall that we had 2 acetyl-CoA molecules, and for each acetyl-CoA, the cell makes 3 NADH, 1 FADH2, and 1 ATP. So now our roster has expanded. We've added 2 more ATP for a total of 4. We now have 10 NADH, and now we have 2 FADH2. So what is FADH2? Like NAD+, flavin adenine dinucleotide, FAD, extracts electrons and protons in the form of hydrogen atoms. Checkpoint 4. In total, how many CO2, ATP, NADH, and FADH2 are produced from the acetyl-CoAs in the citric acid cycle? 2 CO2, 1 ATP, 3 NADH, and 1 FADH2. B, 4 CO2, 2 ATP, 6 NADH, and 2 FADH2. C, 2 CO2, 1 ATP, 3 NADH, and 2 FADH2. Or D, 2 CO2, 1 ATP, 3 NADH, and 1 FADH2. And finally, we have one more step to go, and that is oxidative phosphorylation. This actually occurs along the inner mitochondrial membrane. And this gets to the reason why the inner membrane of mitochondria has so many of these folded structures called cristae. By folding up its membrane, it increases the surface area in order to do more oxidative phosphorylation. The NADH and FADH2 molecules produced in glycolysis, pyruvate oxidation, and the citric acid cycle concentrate in the mitochondrial matrix. Embedded in the membrane, the inner membrane surrounding the matrix, are a number of transmembrane proteins that take the hydrogen atoms away from NADH and FADH2. Now remember, those hydrogen atoms came with electrons, and that's where the name electron transport chain comes from here. NADH and FADH2 molecules that were produced in glycolysis, pyruvate oxidation, and the citric acid cycle all are collected here in the mitochondrial matrix, and we're going to see what happens to them. Okay, so each NADH and FADH2 molecule carries a pair of electrons extracted from the breakdown of glucose. Those electrons are transferred to membrane proteins. We see here 3 of them. So let's begin with NADH. There is a protein called NADH dehydrogenase, which takes the electrons from NADH and uses their energy to pump a proton from the mitochondrial matrix to the inner membrane space. So here we see a hydrogen proton exiting down the bottom. Okay, those 2 electrons there we're not done with them yet. Um, Electrons are transferred from NADH dehydrogenase to a small electron shuttle called ubiquinone, which transfers them to what's called the BC1 complex protein. The BC1 complex protein uses the energy of these electrons to pump another proton from the mitochondrial matrix to the intermembrane space. Electrons are transferred to another small electrolyte shuttle called cytochrome C, which transfers them to the cytochrome oxidase complex. Cytochrome oxidase complex uses the energy of these electrons to pump another proton from the mitochondrial matrix to the intermembrane space, which is why we consume oxygen when we breathe. The oxygen acts as a terminal electron acceptor. So finally, the cytochrome oxidase complex uses the electrons to reduce oxygen combined with protons in the matrix. This transforms oxygen into water. Electrons from FADH2 are passed through the same transport chain but are transferred first to ubiquinone instead of NADH dehydrogenase. And after running through the electron transport chain, now we have a high concentration of protons in the intermembrane space that is built up. All of these NADHs and FADH2s that are passing their protons through these transmembrane proteins builds up a high concentration of them. And what do we know about high concentrations and low concentrations across a semi-permeable membrane is that they will want to balance each other out until there is an equilibrium in concentration across it. So protons diffuse across the inner mitochondrial membrane through an enzyme called ATP synthase. ATP synthase is an incredible machinery. It's a molecular rotary motor, and the flow of protons through the protein causes it to rotate. And like a water wheel, the rotation of the enzyme is used to drive ATP creation. So at the bottom here, we can see that ADP plus phosphate, listed as PI here, creates ATP. For every 10 protons that passes through ATP synthase, 3 ATP are created. Or you can divide the two and say, okay, for every 3.33 protons, you get about 1 ATP. And scientists have been able to capture this ATP synthase protein spinning around here. So these are the gyrations that are caused from the protons going through the ATP synthase, and ATP can be synthesized through this motion. And this is the little secret about aerobic cellular respiration and why multicellular organisms such as us need oxygen. And that's because we require a lot of energy to do our functions. And we saw how much ATP was made from the other cycles or the other steps, not very much. Well, on average, 34 ATP molecules are generated during oxidative phosphorylation per glucose molecule broken down. 34 just from this cycle here, a step here. Checkpoint 5. How many molecules of ATP would be produced from the flow of 50 protons through ATP synthase? Now that we've covered aerobic cellular respiration, let's compare it to its counterpart, anaerobic cellular respiration. Now, let's cover those words there. Aerobic, you've probably heard of aerobic exercise. Aerobic just means that it requires oxygen. So, anaerobic, an means without. So, anaerobic means that it happens in the absence of oxygen. Aerobic cellular respiration includes all four steps that we just finished covering. Anaerobic cellular respiration has one problem, though, and that is it lacks oxygen as a terminal electron acceptor. Without oxygen, the electron transport chain would get backed up and shut down because there would be nowhere for the electrons to go from NADH and FADH2. Here, the top green arrow illustrates how NAD plus is regenerated during the electron transport chain by reducing oxygen. In the process, the beginning can start anew. Glycolysis can start, but it needs NAD plus in order to help with the oxidation process. Okay, so what do we do here without oxygen? And the beautiful thing about aerobic cellular respiration is we're getting so much ATP out of the process here. Okay, the process makes 2 ATP in glycolysis and 36 in the last three steps, mostly from the electron transport chain. Well, fermentation has created a workaround without oxygen to regenerate NAD+, so that glycolysis can continue. Unfortunately, it is much less efficient as far as the amount of ATP produced because only the ATP from glycolysis is generated. And fermentation is what allows us to regenerate that NAD plus so that we can do glycolysis again. So let's take a look at this. Steps involved are fewer and more quickly undertaken in anaerobic conditions. And instead of fully oxidizing glucose into CO2, an organic compound is the terminal electron acceptor. Ethanol fermentation is one type and is a process utilized by organisms such as yeast in bread, wine, and beer. In ethanol fermentation, ethanol and CO2 is formed. We can account for all of the carbons because ethanol has two carbons in it. So two ethanols plus CO2 plus two ethanols plus two CO2 molecules is equal to six carbons, the same number of carbons in glucose. The result of this is only 2 ATP, however. Keep that in mind. Lactic acid fermentation is another type of fermentation. We are probably most familiar with it in yogurt making, and it's responsible for the tangy flavor in yogurt or fermented milk products. Lactic acid is a three carbon molecule. So, can anyone guess what happens with CO2 here? And actually, you might have guessed it, there's no CO2 generated in this type of fermentation. Two lactic acid molecules are produced, and therefore that's six carbons. We don't have any CO2 off gassing. All right, last checkpoint, checkpoint 6. You are a brewer who uses yeast to produce alcohol. What gas must be absent for this to happen? And what gas is produced during its fermentation? Okay, thank you everyone for joining. That is the end of Chapter 7, Cellular Respiration.